Okay, now that we're done with the nucleosome structure for now, where we've talked about how the histones interact with the DNA and the very specific features of these components that allow for a very stable structure to form, I now want to move on to the next section, and that is a section where we will discuss how these nucleosomes will fold upon each other to form the 30 nanometer fiber. Okay, let's get started. Okay, let's go ahead and move on to this next topic of where we're going to talk about going from the nucleosome to the 30 nanometer fiber. After the 30 nanometer fiber, there is more elaborate folding and compression of the chromatin, but not a lot is known about it. So really this is where we're going to stop. And even at this point here, we don't know a whole lot. But let's talk about what we do know. Now, even though we don't know exactly how this 30 nanometer fiber forms, we know it exists. So let's begin with that. The 30 nanometer fiber exists. And we know it exists because of some rather beautiful electromicrograph work showing what appears to be are sort of beads on a string from the nucleosome, like so, and these EMs show the nucleosomes kind of stacking in some way on each other. So kind of the folding of these nucleosomes in some organized manner. So they exist, but what forms them? There are a few models that suggest how they form. Your book talks about some models that have been proposed on how the 30 nanometer fiber forms. However, there are some problems with the model. Now, even though I don't want you to know the specific models, what I do want you to know are the problems with these models. And the problems are all associated with the H1 histone. Now remember, I told you before there were some disputes about whether the H1 histone was important or what its exact role was. But most believe it has some function in binding to the linker DNA and connecting the nucleosomes, and others believe it actually has an important role in forming this 30 nanometer fiber. So the problems with the models are, first, some of the models were proposed doing experiments where there was an absence of H1. And then some of the models were proposed doing experiments with extra H1, as opposed to using a physiological amount of H1. Some of the reasons why we believe the H1 histone is important is from work done with mice. Mice have 6 H1 genes. 6 genes that code for this H1 histone. Gene knockouts, where you remove the gene from the mouse genome. So gene knockouts of H1 of individual. So gene knockouts of single H1 genes changes expression level, suggesting it's important. So the next experiment really proved the importance of H1, I believe. And that is if you take and knock out 3 of the 6 H1 genes, the mouse dies as an embryo. So it's embryonic lethal, suggesting that these are really important. And when we want to propose models, then it's probably a pretty good idea to include them in those models. Okay, there was one other problem with this model, and that is when they proposed these models, it was done assuming linker DNA was equal. Now we know that the DNA within the nucleosome is consistent. There's very little variability there, but there's quite a bit of variability in these linker DNAs. So the models did not take that into consideration. This variability here is seen when we measure this nucleosome repeat length from here. And when we measure that, we can see ranges from 165 to 196. When you're doing structural studies, these make a very big difference when you're trying to figure out the bins of the nucleosomes between each other. So over here, let's just write down quickly here what is known about forming the 30 nanometer fiber. So what is known? Well, we know what's important. We know that the H1 is important. How it's important, we don't know yet, but it's important in forming this 30 nanometer fiber. We also know that the nucleosome repeat length is also important, or more specifically, that linking DNA. is very important. In addition, scientists are beginning to find certain proteins that they believe are important in forming this 30 nanometer fiber. And your book talks about a few of them, and I really want you to focus on just this one. It's called polycomb repressive complex 1. Or PRC1. PRC1 has a long history, and its rise to fame came in Drosophila, where it was associated with repressing certain genes needed for embryonic development of Drosophila. But what has been shown more recently, that if you take nucleosome structured chromatin and you add with it PRC, you get the 30 nanometer form. Where PRC1 is binding to these nucleosomes, that is these beads on a string, to form this is not clear. And there are what I'm just going to call other proteins. Still learning a lot about this, but you should know that the H1 is important, that the nucleosome repeating length is important, how far they're spread apart, the nucleosomes, and this one protein called polycomb repressive complex 1. Okay, I want to return to some terms we talked about before. Euchromatin and heterochromatin. Remember, we said if you had a piece of DNA, or let's just draw a whole chromosome here. We have our centromere here. And just to remind you, in case you forgot, the centromere is important in attaching spindle fibers during meiosis and mitosis. We also have these ends down here called telomeres, and they help stabilize the ends of linear chromosomes. And we said there are parts of this chromosome that are be classified as heterochromatin, and I'm going to just draw them like this, where the DNA in these regions are very compacted. And we know that these areas of heterochromatin are transcriptionally inactive. If there's genes here, they're not going to be turned on. I'm just going to put an X over them. What I don't think I said when we introduced this topic before is that heterochromatin is found always near the centromere and always near the telomere, in addition to other areas of the chromatin here where there are areas of transcription repression. So heterochromatin is shown by an X. Whereas euchromatin, I'm going to show with an arrow, are all these areas that are not compressed. And these areas are transcriptionally active. There are never genes found near the telomere or next to the centromere, because they're always heterochrome. But these areas here, depending on the tissue and the species and the time of development, some areas are heterochromatin and some areas are euchromatin. And there is a degree of back and forth between these two structures. Now, there are two terms that I didn't say or mention when we talked about this, and they are constitutive heterochromatin and facultative heterochromatin. Constitutive heterochromatin would include the centromere and the telomere, and areas where there are satellite DNA. Satellite DNA we can for now define as areas of highly repetitive DNA. They don't contain genes. Facultative heterochromatin, however, are areas of the chromosome, they might be these regions here, where in some tissues or developmental periods they are heterochromatin, and in others they are euchromatin. So for facultative heterochromatin, whether it's heterochromatin or euchromatin, is dependent on tissue, development, and I'm going to put and stress. So certain environmental pressures could change a region from heterochromatin to euchromatin or vice versa. I want to bring back bar bodies for just a little bit and remind you that these are the inactive X chromosomes, and they provide a great example of what a facultative heterochromatin is. So let me explain this a little bit better. in individuals who have two X chromosomes, all their cells have two X chromosomes. However, in some cells, one of them is formed into a bar body, which I'm just showing as a circle there. And in other cells, the other X chromosome is formed into a bar body. This is a good example of facultative heterochromatin, because in some cells, the heterochromatin, which is the bar body, is on one X chromosome, and in other cells, it's the other X chromosome that is formed into heterochromatin, or the bar body, as I said before. The reason many organisms do this, particularly mammals, is to compensate for two X chromosomes. We want to make sure males and females all produce one value of X chromosome. And so the way we do this is by having, in individuals with two X chromosomes, having one of those X chromosomes inactivated. As we move forward with this next chapter and the chapters to come after, a key question we're going to have is when we have genes, which I'll show here in green, that is in areas that can't be expressed because they're tied up in these nucleosomes, how is it that we can turn those genes on? What do we have to do to unpackage that nucleosome, to remove those histones, so that we have access to those genes? And so that's what we're going to be talking about next, is how do we gain access to genes that are trapped in heterochromatin. Okay, that's all I have to say in this podcast, or these past two podcasts. So a quick summary here. In these podcasts, we focused on the chromatin. And more specifically, we focused on the packing of that chromosome, or the chromatin. We focused primarily on the nucleosome. That's where we spent most of our time. Again, because that's what we know most about. We talked about those special interactions between histones and other histones, using those histone folds. We talked about how histones interact with DNA and how features of the DNA sequence and the phosphate backbone are important in interacting with the histones. And if you remember, I really stressed the positive amino acids that are found on these histones and the negative charge on the DNA, how that's a very important interaction. Don't forget that. We also talked about the importance of these histone chaperones in establishing histone-histone interactions and forming the nucleosome. We then talked about the 30 nanometer structure, or fiber we'll call it. In this we talked about the importance of the nucleoid repeat length and the H1. We also talked about the PRC1 protein and how it's likely involved in forming this 30 nanometer fiber. And then we closed by talking once again about euchromatin and heterochromatin. But this time we introduced some new terms: facultative heterochromatin and constitutive heterochromatin. And I ended with talking about how we're going to move on to the next chapters. How it's important to know how to unpackage these chromatin so we have access to specific genes. And then how to make sure we package them back up so that we can hide other genes so they can't be expressed. Alright, if you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.